Human beings are incredibly complex. Something as simple as even just saying this sentence right now requires me to breathe in oxygen into my lungs, which is then transferred to my blood, which is then carried up to my brain when my thoughts develop. And from there, the signal gets relayed down to my muscles, enabling my mouth to move and so I can talk. Now, all of the different cells involved in that pathway are specialized to do their different functions. But what they have in common is that they've actually come from the same type of cell, a stem cell. The developing embryo contains stem cells that become more specialized to give rise to the entire human body. But sometimes errors can happen in that process. For example, cells might get stuck on their way towards becoming a cell of the lung or the liver or the blood, but still continue to multiply and grow where they are, kind of like a pileup on the motorway. But in the human body, this can lead to cancer because the cells that are multiplying aren't behaving as they normally should. And that is exactly what happens in this type of childhood blood cancer that I'm studying. We know that children diagnosed with this blood cancer have multiple different errors present in their cells, but we don't know how exactly each of these individual mistakes contributes to the overall picture of cancer development. And that's what I'm trying to figure out with my PhD. Now, what's unique about this particular blood cancer is that it actually originates before birth, but we can't take cells from a fetus. And so the only way to really study that type of cancer in the lab is by using a special type of stem cell that I have. I'm genetically engineering these stem cells with the errors or mistakes that we know are necessary for the development of this blood cancer, with the aim of trying to understand exactly how and why each of these individual errors turns what would have been an otherwise healthy blood cell cancerous instead. From the work I've done so far, I've determined that one of these errors I'm investigating actually does affect that normal process of blood development, but I know it's not enough to result in blood cancer. Work is ongoing to determine what additional errors are necessary for that full-on blood cancer progression. But why am I doing this research in the first place? Well, that's because currently, the only treatment available for children with this type of cancer is chemotherapy, which, as we all know, has horrific side effects: heart toxicity and early death, and an increased risk of getting additional cancer, to name a few. Chemotherapy does that because it can't tell the difference between our normal healthy cells and the cancerous ones, so it just wipes everything out. With the model I'm developing, I'm hoping to determine the exact errors that are necessary for the development of this blood cancer, allowing us to then design drugs that can specifically target those. This will mean that we'll be able to kill off the cancerous cells without harming our normal healthy ones. Kind of like only removing the defective toys on a factory conveyor belt as opposed to just aborting the whole production. Meaning that one day, hopefully, children will no longer have to endure horrific treatment in trying to cure their blood cancer.